I'd like to welcome you to the fourth annual John C. Martin Memorial Lectureship. We're here to remember John Martin. For those of you who don't know John, John was an amazing scientist. He really was the person who pioneered nucleoside antivirals, which have had a huge impact on everybody. And he was an extraordinary businessman. And he was the person who really built Gilead into the powerhouse it is today and the powerhouse it is in the antiviral space. But he was also, on top of that, a really great humanitarian, because he was really one of the first people, if not the first person, who really kind of advocated to supply low-cost antiviral drugs to low- and middle-income countries. And so, as a result, I think the drugs developed at Gilead under John's leadership and his efforts to make those drugs available to the rest of the world have saved millions and millions of lives. So there's not many people who can say they've done as much. John was also a great friend to Scripps. John, as he was stepping down from Gilead, decided to join the Scripps board. And, given all of our efforts to really bridge basic and translational research, John's advice and insights as a board member were really invaluable. This is actually, I think, this would have been John's 74th birthday today, okay, somewhat serendipitously. And it's amazing how many times I'm sitting around a room and we're thinking about something and somebody says, too bad John's not here, because it would have been great to see what he thinks about this, okay? And it happens a lot. John's also continuing to have an impact on Scripps, okay? We just had 3 graduate student fellowships given to us by the Martin Foundation, which is wonderful, because if it turns out this year a lot of graduate schools have stopped taking any graduate students, okay? And those fellowships allow us to continue to have a graduate program, a robust graduate program. So I think we all owe John a lot. And even nowadays, as we think, I wonder what John would have thought about that. As we think about the change occurring, and I could characterize it as craziness in Washington regarding science and science funding and science policy. I think what we're missing is somebody who understands the value of basic research, who understands not only the impact of basic research on public health, but also on the economy, and understands policy and governmental policy. I think we're really missing John as somebody who could actually speak for science and medicine in the current environment. So I'll stop there. If we could put on the agenda slide. Thomas will make the introductory remarks, and then Steve McKnight will give a lecture. And then Gregg Alton will follow up with comments on John and Gilead. And then there'll be a reception outside. And I'll start by introducing Thomas. Thomas is actually the vice president of virology at Gilead. And, as I said, Gilead is probably the pioneer in the development of many antivirals, HIV, HCV, okay. Thomas himself actually played a major part in the development of remdesivir, which we all know was the first small molecule drug used for the treatment of COVID-19. And also, more recently, lenacapavir, which is probably the most exciting advance in the development of antivirals, probably in the last 30 or 40 years. So, and we also collaborate with Thomas's group and Gilead with the development of other HIV antivirals. So we know Thomas well, and we really appreciate both his science and his collaboration. So, Thomas, I'll turn it over to you for the start.
Good afternoon, everybody, and thank you for the kind introduction, Pete. Thanks very much for this opportunity to say a few words about John. It's a real honor and it's absolute pleasure to be able to speak about John. And for many years, you know, he was a big figure for me somewhere at the leadership building. But over the years, I think I had the opportunity to know him better, connect with him, and actually understand why he's doing things the way he's doing them. And I think for today, you know, we can talk about how John travels and jokes and his quotes and stuff, but I thought, given the situation in the world around us, I really want to make one story clear, and his dedication to the partnerships and collaborations as the engine that really drives the innovation and progress. You know, for me, particularly in the antiviral medicines, because that's what I've seen, and I had a fortune to be close to that and a part of it. So the story I want to talk about a little bit is, many of you know about it or lived it even here. For some of you, it's new, and it really started in the middle of 1980s with this seminal publication in Nature, which was the collaboration of two academic institutions, a Catholic University in Leuven in Belgium and the IOCB Institute in Prague. And the two groups were the virologists from Leuven and the chemists from Prague, led by Eric de Klerk and Tony Holly respectively. And in this seminal paper, they basically came up with a new class of the antiviral nucleotides, which the uniqueness of that is captured there a little bit on the structure, because it has one phosphate or phosphonate group that is covalently attached to the nucleoside body. And John was at that time very well familiar with the class of the nucleoside analogs, but this was quite new because it brought a little bit more stability, faster metabolism, and longer half-life of the active triphosphate metabolites of the nucleotides. So originally, this invention was in the center of attention for Bristol Myers, where John worked at in late 80s, but ultimately Bristol, for business reasons, they let it go, returned the invention back to KU Leuven and IOCB. But then John moved to Gilead, and in the end, thanks to John's vision and intuition, these were licensed in 1991. You know, there is a kind of amusing piece about the whole early history of Gilead, and it's actually a play that was introduced in small theater in Prague in 2018 called The Elegance of the Molecule, and this is talking really about the new class of the nucleotide antivirals. And here you see the one scene that particularly is devoted to the negotiation of the licensing agreement between KU Leuven, represented by Eric de Klerk, IOCB by Tony Holly, and Gilead by Michael Riordan, the CEO at the time, and John Martin, who was the head of R&D at Gilead, and freshly coming from the Bristol Myers. And so in this scene, John is saying to Tony, who is worried about where all the money will come from and who is going to take all the risk. So he's saying, Tony, we will put the chemists back in the game and we believe in your intuition. Which I think it's true to the core of the John, because he was talking maybe about Tony's intuition, but I think he was talking primarily about his own intuition and vision that this new class of the molecules could actually be developed into meaningful drugs and make the impact around the world. And just to prevent all the confusion, these are how the real people actually looked like. And I can tell you from my experience, neither Tony Holly nor Eric de Klerk ever sported any facial hair in their life. So it's really the actors who took the artistic license. and it actually worked out really nicely. And so this was 1996. The picture was taken, I think, in Leuven, shortly after the first antiviral product was approved, Cidofovir, for the treatment of CMV retinitis. And it was also the year of the transition of the CEO function from the Michael Riordan, who was the founder of the company since 1987. And he departed and passed the baton to John in 1996. So John, Tony, and Eric were lifetime friends and partners. And I think I like to emphasize this because this really reflects one of the parts of the John's generous personality is to stay connected with the friends and the partners for the long time or lifetime. And, you know, they met regularly at scientific conferences. That's one from 1995 in New Mexico in ICAR conference. Then John was always clear and very focused on the recognition of Eric and Tony. So the 2003 picture is actually, I think, from Board of Director meeting in California where the board recognized Tony and Eric for their contribution to the antivirals. And then moving to 2011, that was the 75th birthday of Tony Holley. At that point, Tony was quite ill and aging. And it was also 25th anniversary of the seminal publication that I showed on the first slide. So there was quite a gathering, actually, that was organized in Prague, and John was very closely involved in organizing it. And he asked me to help him. And so I had a weekly meetings with John to basically plan this event and understand what we are going to do there, how we are going to organize the symposium. I have never had the weekly meetings with John before and after. But this was very important for him, and he has been doing it for Tony. And this was the last time when the three of them actually met when all were alive. And there was one sort of the figurative gathering of all three of them, and it was in 2015, and it's also the picture from Prague, from IOCB, when the bust of the Tony was installed at IOCB New Chemistry Building, and John and Eric came, and then, you know, figuratively, all three of them stood there together. And so this partnership of the scientist and the entrepreneur scientist actually was hugely impactful. And when you speak about the best nucleotide from the class that has been licensed to Gilead, tenofovir is a part of the 14 different antiviral products for different indications. And so I try to go to literature and try to figure out, is there any other example when one single molecule in two different forms, in two different prodrugs, actually is the part of this large portfolio of the different molecules. And as you see, this moved over time, and it started with the single agent Viread, which is the first generation tenofovir prodrug, and moved into more and more complex single tablet regimens that ultimately culminated with Biktarvy, which is the best-selling, most frequently used HIV drug worldwide. So you see the genesis of the innovation across the year and improving the products one step at a time to serve more patients. The whole story is really nicely covered in this small book called Cold War Triangle that was read by or written by the Renil Deluxe, who used to be the ambassador of Belgium to Prague in early 2000s. And so she frequently actually made friends with Eric, John, and Tony, and frequently met with them when they came to Prague. And the Cold War Triangle, really what she meant by that is the three different places geographically that form the triangle since the late 80s, basically, which was at the time when there was still the, Iron Curtain and the Eastern Bloc was going strong. And John was the visionary person who basically crossed the boundaries. He didn't care about the Iron Curtain, and he went to Eastern Europe and looked for the new antivirals there. When you look at the map, it actually looks more like a bridge, right? It doesn't really look as a triangle. And I think this is highly symbolic in these days. It's really a bridge across the continents. It is the bridge between the basic science, represented by the virologist in Leuven and the chemists in Prague, and the applied pharmaceutical science by John and Gilead. And this connection really was the successful, synergistic, and was most important for the building the foundation of Gilead. John was very fond of Prague, and he liked it not just as a city, but he also was very adamant and focused on helping the IOCB and helping the scientists there. So you see the picture there from 1996 when Tony Holley was still director of the IOCB. Then 10 years later, at the 70th birthday of Tony, in the same year, we realized that John's idea to actually form, together with Bill, Laurie, and a number of the people from Gilead, Swami, to form the Gilead Sciences and IOCB Research Center, which basically is renewed on the 5-year cycle, and it's still going, and it's going to be on 20 years trajectory next year. 2011, you actually see there the signing the agreement, extension agreement with then director of IOCB's, Zdeněk Havlas. And the last time when I had opportunity to travel to Prague with John was in 2019, and that picture is actually taken from the Tony Holley's office, which they turned into sort of a small museum. And it was very symbolic when we gathered there, actually, and I think a lot of memories, and John spent a lot of time in that office talking with Tony about the chemistry. And it was also the time when John and the others from Gilead came to see the play, The Elegance of the Molecule. So this was the sort of last chapter for me. November 7, 2019, meeting in Palo Alto. That was the last time when I met John in person. Then COVID came just a few months after that. It became a lot more complicated to meet in person. And early 21, unfortunately, John passed tragically and very unexpectedly away. But this is important also from the sense that this was the brainstorming session actually to help the IOCB, so there are 2 gentlemen from IOCB leadership there, to see what can be done there more in terms of the translational research and the drug discovery. And you see the Pete there also, and the reason why he is there is actually one of the idea was to form the Translational Institute in Prague that would be modeled based on the caliber here at Scripps a little bit, which eventually actually happened. And that institute with the name Pharmtheon actually opened the door last year. It has more than 20 scientists, and Bill is now involved on their board. And so this is really amazing because John was out of Gilead at that time. He was focused on his foundation, but he still thought about his friends in Prague and how to help them. When I think about the history, there is one moment or one time which I think is very symbolic actually, and this is this picture of the many luminaries of the antiviral research and many role models for me at least, and this was my first international conference of antiviral research, or ICAR, in 1997 in Atlanta. And this picture was basically taken at the time when the lady Trudy Elion was, that was her last meeting. She was very active as a part of the Antiviral Society. And she was also the Nobel laureate for the first examples of the rational drug design. And one of the drugs was acyclovir. And Trudy was really good nucleoside chemist. She was the chemist by the training. And working at the Burroughs Wellcome, the expertise on the nucleoside really helped to actually advance the field. And I cannot help myself looking at John as a fresh CEO. It was kind of passing the baton from one nucleoside chemist to another and carrying the flag for the antivirals. And I cannot imagine none of these people who were gathered here actually, I don't think that they could have ever thought what would came out of that, what would came of John's leadership in terms of his legacy, the legacy of Gilead for the innovation of antivirals. So you see here all the antiviral drugs that have been discovered or developed at Gilead over the course of almost 3 decades, and it's 23 different products, with the last one being the lenacapavir, which John was this last year CEO when lenacapavir was actually discovered and declared as the development candidate. And I had the fortune to be involved in that, and I think we applied the exact same model for the close connection and the collaboration between basic science and the applied drug discovery research. The basic science came from the HIV basic field, understanding the function of HIV capsid in HIV replication cycle. And then we were able to appraise the data and make the decision that this might be the very exciting new target. And 10 years later, the lenacapavir came out of that. I mean, we can count the number of the drugs But what really matters is the impact on the people around the world. And here you see some numbers, and it's staggering millions who benefited from HIV drug, HCV drug, and most lately from remdesivir and COVID-19. And this, to a large extent, as Pete said, was thanks to John's vision to actually be able to find the way how to make the drugs accessible to the people in the need in the low and middle income countries. And you will hear from Gregg Alton about a lot more detail about how the whole access system was actually built. So John, happy birthday today. I didn't know it's exactly the day. If you can see us, I really want to thank you for your leadership, for your vision, for mentorship and innovation. And I think at least for me, and I'm sure for many people here who knew John closely, you have been the beacon and North Star in our lives, both scientifically and personally. And I think we need that example these days when things got a lot more complicated than we have ever expected. Thank you so much.
Hello, I'm Benjamin Cravat from the Department of Chemistry, and it's my great honor to introduce Dr. Stephen McKnight, a distinguished chair in basic biomedical research at the University of Texas Southwestern. By any and all accounts, Steve is a towering figure in the fields of biochemistry, chemical and molecular biology, and cell biology, and I could gladly stand up here for hours and recount his dizzying array of transformative research discoveries that range from defining promoters as the fundamental regulatory sequences in our genome to identifying novel families of transcription factors, and through doing so, pervasive protein folds such as the basic leucine zipper, to elucidating the mechanisms by which our cells adapt to oxygen stress, and then to the point of mapping low sequencing complexity domains in proteins as the basis for self-associated mediated phase separation in cells. But I thought I would do something different and a bit more personal in this introduction, and instead share with you the positive impact that Steve has had on my career and others in the Scripps community. In addition to the Martin Symposium, we have a handful of other extremely special lectureships at Scripps, one of the longest running of which is this distinguished lecture series organized by the graduate students. In this series, the speaker is directly hosted and spends most of their time during the day interacting with students. As a graduate student at Scripps in the mid-1990s, I nominated and hosted one speaker for the Distinguished Lecture Series, Dr. Stephen McKnight. At that time, I was still in the process of constructing my personal understanding of the field of chemical biology, but I had a visceral sense that some of its most innovative and impactful outcomes may reside at the interface of academia and biotechnology. Steve had, a few years back, moved from an academic position to lead research at a startup company, Telluric, with the ambitious goal of developing drugs for transcription factors. Well, today, it may not seem so unusual for an esteemed academic researcher to move to a biotechnology company. I can say that in the early 1990s, it was about the furthest thing from typical. After spending the day with Steve at Scripps, it became obvious why such a move wouldn't faze him. For Steve, it was all about finding a path where he could have the most profound and lasting impact on science, discovery, and human health. As a young scientist at the time, and also a student in a new and still unaccredited graduate program that most of those in the country had never heard of, I found Steve's independent spirit and complete disinterest in the rituals of traditional academia invigorating. I learned from Steve to trust and follow my passion, regardless of whether it aligned with convention. Shortly thereafter, Steve blazed another incredible trail, this time to Dallas, where he joined UT Southwestern to lead their biochemistry department in exciting new directions that emphasize the importance of chemistry as a key discipline for understanding and ultimately controlling biological processes. It was there that Steve's second major touch point with Scripps emerged. My roommate in graduate school was an incredibly talented scientist named Rick Bruick. For his postdoctoral studies, Rick joined Steve's lab at UT Southwestern, and together they mapped a key enzymatic component in the HIF pathway that our cells use to sense and adapt to changes in oxygen availability. Interestingly, this same pathway is dysregulated in a syndrome called von Hippel-Lindau disease, or VHL disease, that leads to devastating forms of kidney cancer. Steve generously supported Rick's transition to an independent professorship at UT Southwestern, where they proceeded to collaborate together, culminating in the formation of a biotechnology company, Peloton Therapeutics, notably located in Dallas, that developed the first drug, belzutifan, for treating VHL disease-related renal cell carcinoma. Peloton was later acquired by Merck, who now sells the drug under the brand name Welireg. I still keep in touch with Rick and asked if he might share a few thoughts about Steve. So these are Rick's words. Steve has never been one to chase in vogue fields or tools. He never met a wild hair idea that he didn't like. The crazier, the better. Of course, not all of them hit, but when they did, they led to major advances. I, Rick, was fortunate to get swept up in one of Steve's early bets at UT Southwestern to identify and characterize mammalian transcription factors containing PAS domains on the hunch that they would be druggable. This was the seed from which our work to drug HIF2 was born. Along the way, we were enabled by Steve's vision to bring biologists, chemists, and structural biologists together under one roof, not unlike Rick's in my early days here at Scripps. Rick then said later on, Steve would assemble the pieces to form Peloton to translate our proof of concept discoveries into belzutifan, a HIF2 transcription factor antagonist that is now approved to treat VHL diseases and clear cell renal carcinoma. That's Steve in a nutshell, according to Rick. Chase a unique vision, pave a way, and make a discovery. Like Rick, I too see many parallels between Steve's role at UT Southwestern and Richard Lerner and Pete Schultz's impact here at Scripps. These are rare individuals who possess the creativity, intellect, and drive to change the world. Each could have decided to exclusively focus on building their personal careers, but instead pursued paths for the greater benefit of our scientific community and humanity in general. I can thus think of no one more appropriate than Steve McKnight to give the Martin Lectureship here at Scripps Research. Steve, thank you for all you have done for science and human health, and we look forward to your lecture today.
Ben, that's hard to follow. It's an absolute delight to have been invited to give this lecture, and I don't know who was responsible for the invitation. 30 years ago, it was Ben who invited me to give a seminar at Scripps, but this one, I'm not sure. Pete. Well, thank you. It's quite an honor to be here. And kind of the first thing that I did upon receiving this invitation was to Google John. And I was blown away. I knew that Gilead was a terrifically successful company. I knew that, but I didn't know who was behind it, who made the magic happen. And you'll hear much more about John, but I will say this. Among his many, many accomplishments, I think it's absolutely outrageous that he was able to form a consortium with other companies to put drugs together for the triple therapy for HIV. For-profit companies are not known for being generous and taking risks, but this was unbelievably important for patients suffering from AIDS. So what a remarkable person, and to be able to give a lecture in his honor is very much an honor for me. So that's the first thing I wanted to say. The second thing I wanted to say is that I long ago looked at Scripps when Richard Lerner was the president, and I wondered, why would he want to infuse so much chemistry into it? Because I'm a biologist, I'm not a chemist, and I thought it was kind of strange when the biological fields with gene cloning and all of molecular biology on fire, why would someone want to put chemistry into the mix? And so I watched that and I couldn't understand it. And then I watched Pete do the same thing. And somehow that inspiration somehow got to me. So when I had my chance to become a leader, an academic leader at UT Southwestern in the biochemistry department, I did exactly the same thing. I hired chemists, and I brought chemists into the fold, and it was probably the single best thing that I did to create a unique environment of people chasing ideas that were unique and different from the norm. So the last thing I'll say before starting my presentation was that when I was a kid, I grew up in El Paso, Texas. I was born in 1949, so in the late 1950s, my family drove across the desert to La Jolla. And I was maybe 10 years old, and when you've grown up in the desert of El Paso, to come to La Jolla, California, it was like coming to heaven. It was unbelievable. And I'll just tell you a couple of things that I remember very well. One thing is I remember going to an amusement park called Belmont Park, I think. I don't know if it's still here. But that was so fantastic for a little kid. So I remember Belmont Park. I remember body surfing at Wind and Sea Beach, where we had a little apartment. I remember Anthony's Fish Grotto, and it's probably long gone. But I also remember my parents taking me to the Oceanographic Institute, Scripps. And so here I am, a 9 or 10-year-old kid, and to me, Scripps, that name was like, whoa, that's walk on water. That's the really good stuff. And to think that here I am as a 75-year-old man being able to come and give you all a talk about my research, it was not what I imagined for my life. That's all I'll say. Okay, I'm going to talk about research over the last 10 or 15 years in my lab that is very much of a basic scientific nature. Maybe towards the end, I might talk about some aspects of the work that I think could be relevant to human health and disease, but by and large, this is just a scientist chasing his nose. We're trying to find unique things in biological sciences and chase them for the fun of the adventure of trying to figure out how biology works, how nature works. And it goes back 40 years. So I was a young scientist, and at the time I was competing with Bob Rader and Bob Tejan and Phil Sharp to purify the first ever transcription factor. And so many of you in the audience were not even born then. So this is a long, long time ago. But we were able to purify what we call gene-specific transcription factors. And in studying them, we were able to clone their encoding genes, sequence them, and really begin to understand what was a transcription factor, how were genes switched on and off. And so what we were able to sort out fairly rapidly was that they are composed of two domains. One domain was responsible for the specificity of binding to the right sequence of DNA, to hook onto the right gene. So that's what we call the DNA binding domain, and we were able to sort this out because Max Perutz and John Kendrew and the icons of structural biology had taught us how to do crystallography, so we were able to solve the structure and understand how the structure, how the form specified function. And so we solved the structures of these DNA binding domains, and they came in various flavors, homeoboxes and zinc fingers, and the one I studied was a leucine zipper. So we sorted this out, but that was not the whole story. In addition, these transcription factors had another domain that was required to activate transcription. So it was not good enough for the protein to bind to a gene. It needed to tickle the gene to cause the gene to activate and cause the RNA polymerase to be attracted to the gene and activate transcription. So we call this the activation domain. And if you lop this off, if you clip this off so that you only had the DNA binding domain, it was neutered. It could still bind to the gene, but it couldn't activate transcription. So we foresaw two different roles, gene binding specificity and this activation process. And this process over here, we did not solve. We worked on it. We were really interested. The one I worked on was a protein called VP16, and lots of citations, lots of great work, but we never sorted it out. And the reason we didn't sort it out is that this part of the protein would not adopt a stable structural fold. It was disordered. And so these were disordered sequences, and we couldn't figure it out. Now, I had a talk this morning with one of your esteemed faculty members, Peter Wright, and Peter Wright is still studying this to this day, and he's doing fantastic work on it. So it's taken 40 years, but there's progress being made. But we flood this. We didn't get anywhere on it. And why is it that this is ordered and this is disordered? Well, the reason for that is this has complexity and this lacks complexity. And what do I mean by that? I'm showing you here the sequence of one of these disordered domains that we've studied intensively for the last decade, and it's a protein called fused in sarcoma, or FUSE. And it's an example of an intrinsically disordered low complexity domain. So what I mean by low complexity, if I look at the amino acids that are present out of this 214 residue protein, 83% of them consist of only 4 types of amino acids: glycine, serine, glutamine, and tyrosine. And there's 0 examples of like these 10 amino acids. So the code that is being used by this protein is more like DNA than it is like protein. It's primarily using 4 letters instead of all 20. A normal protein that folds properly typically uses a reasonable distribution of all 20 amino acids. And so I refer to these as low complexity domains because their sequence is kind of not using the full card deck, but is using only a small number. And what that means is the polymer has less chemical diversity. And to fold into a structure, an appropriate structure, chemical diversity is extremely valuable. So these were kind of unique observations, but what happened is in the ensuing years, it became clear that these protein domains of low sequence complexity are everywhere. There are lots of these. They're not just in transcription factors. They're all over the place. Every part of the cell, proteins in every part of the cell utilize these low complexity domains. And so they're all over the place. So here are a couple of points. These low complexity domains constitute around 25% of eukaryotic proteomes. And they're found in thousands of nuclear and cytoplasmic proteins, so they're everywhere. 70 or 80% of all forms of post-transcriptional modification, including phosphorylation, acetylation, methylation. So the sorts of chemical changes put onto proteins, 80% of those are on this 25%. So biology is picking on these. It's using these to execute regulatory functions. And likewise, 80% of all forms of pre-messenger RNA alternative splicing are localized to these low complexity domains. So although they're only a quarter of our proteins, they're really where the action is for regulation. And they've been hard to study because they do not assume structures. So they're not easy to sort out. They're really important, but they're very hard to study. So I was working on these 40 years ago. I didn't solve them. And then out of the blue, something happened about 12 or 15 years ago, and that's shown on this next slide. And that is, I got a surprise that this fused in sarcoma protein formed a gel. And this is the tyrosine glycine rich region of fuse, and it, in the test tube, forms a gel. And the same observation had been made for a different low complexity sequence by Dirk Gorlich and his colleagues at the Max Planck in Göttingen, Germany. So this is another low complexity domain. They saw the same thing. So in working with proteins all these years, I'd never seen a gel. I'd seen lots of things happen, but I hadn't seen this happen. So I said, whatever it is, I'm interested, sign me up. And I wanted to know, of course, what was the basis of this gelation phenomenon, this phase separation phenomenon. And so what I did, having been trained by Oscar Miller, an electron microscopist, I simply put an electron microscope grid on the surface of the gel, peeled it off, negative stained it, and looked to see what the surface of the gel looked like. And what you see in the next slide is an image, a negative stain image of what the gel looked like. And as I think you can see, the gel was composed of gorgeous, uniform polymers. So it's just full of polymers. So the gel is just absolutely full of gorgeous polymers. So clearly what had happened is this soluble low complexity domain of the fused and sarcoma protein went from a solution state where it was a monomer into a polymer. And we could lyophilize the gel, put it in the X-ray beam, and see diffraction rings at 4.6 or 4.7 angstroms and 10 angstroms. And between making this observation of these polymers and seeing this diffraction ring, we knew right away what we had. And what we had was an amyloid-like filament. This protein was making an amyloid-like polymer. And amyloids, as taught to us by the great chemist Linus Pauling, are held together by the zippering up of two extended strands of protein. So what I'm showing here is the canonical beta-pleated sheet organization of two protein molecules, two protein chains zippered together by hydrogen bonding. So the peptide nitrogen of one strand forms a hydrogen bond with the carbonyl oxygen in the other strand, and then you walk your way down the chain of amino acids, and this happens over and over and over, and this is one of Pauling's two genius discoveries about protein secondary structure, the other being the alpha helix. So this is what we clearly had, because this distance is 4.6 or 4.7 angstroms. So we had polymers held together in this way. Now that was the good news. The bad news was that these kinds of polymers are always terrible for biology. They're prions. They're very stable, and they're disaster. You're never going to build biology on this. So this was an exciting observation, but at the same time was very disconcerting. So we did a very simple experiment to compare these polymers with known pathogenic polymers, or very stable polymers. And the ones that we used were SUP35 polymers formed from a yeast protein that's known to form a prion. This is beautiful work by Reed Wichter. And so what you're seeing here is an agarose gel where we've electrophoresed the polymers of this SUP35 protein, and after exposure to different amounts of a denaturant, sodium diacetyl sulfate. So these polymers are resistant to 0.5% SDS, 1%, 2% SDS. The polymers are stable as can be, and this is characteristic of prions. They're rock solid, and they're pathological. They're very bad. When we did the same experiment with our polymers formed from this fused low complexity domain, the polymers dissolved even as soon as we put the loading buffer on. So morphologically, they looked just like prions, but clearly they were very unstable. So there was a difference. So how are we going to solve the difference? And the only way to solve the difference, quite obviously, is to get the molecular structure of these polymers and look in detail at the chemical features of the structure. So we did that in collaboration with a terrific solid-state NMR spectroscopist at the NIH by the name of Rob Tico. And the molecular structure is shown here. So I just want to say three things about the structure. One is it's right here, and it's a small part of the protein. It's not the whole protein. It's just a small part. The second point I want to make is it's always the same part. If I make polymers this week, I get this structure. If I make polymers 2 months later, I get the same structure. That's what's called following Christian Anfinson's rules. The protein knows what to make, and it always makes the same thing. And the third point I'd like to make is that the way the polymers are organized is these beta sheets are in register. So here's amino acid 1 of each of the proteins, and if you follow your way down, so each of them begin to form this polymeric structure at the exact same amino acid. So the amino acid sequence of the blue, green, and red strands are aligned all the way through the structure. So that's called in-register conformation. So those are the three things that we learned. But what did they say about why these are unstable compared with dangerous prions? And so what I'm going to show on the next slide is just a slice where we're looking down the axis of this polymer, we're just looking at the surface of one protomer. So here we are. This is the slice of looking at one protomer of our fuse polymeric structure that follows the Christian-Amundsen rules. And what we look for is hydrophobic amino acids. In this whole interface, there's only one hydrophobic amino acid, and it's a proline. And prolines are not heavily hydrophobic compared with leucines and methionines and so on. And furthermore, as you'll see later in the talk, prolines fight against this structure, as I'll show you. Now let's look at a dangerous prion that causes Parkinson's disease, and this is alpha-synuclein. And it's a very similar structure. It's a cross-beta structure. But the difference is this protomer interface has 23 hydrophobic amino acids, leucines, isoleucines, valines, methionines. So this structure has hydrophobic glue holding subunits together. This one has no glue. So right away we can see, aha, the difference is very clear. One has a hydrophobic interface that holds protomers very tightly together. The other doesn't. So this is not a highly stable structure and offered the chance that we're on to something that might be telling us something about how these domains of low sequence complexity work. In other words, perhaps they coalesce in this sort of way as part of their role in helping biology work. So the problem with biochemistry is that you can do anything in a test tube. You can make anything happen that you want to happen. And the question is, what you're doing in the test tube, is that accurate as to what happens in the cell? That's the challenge of biochemistry. And if you're not careful, you'll get it wrong. And I've gotten it wrong many times. So you have to be careful. So how do you know what you're studying in the test tube is of biological relevance? And Mike Brown and Joe Goldstein and Al Gilman told me, Steve, there's two ways to do this. One is by pharmacology, and the other is by genetics. So I'm going to tell you pharmacology first and genetics second. All right, what about pharmacology? So on the side of pharmacology, we have some chemicals to work with. And these are two aliphatic alcohols, one called 1,6-hexanediol and the other 2,5-hexanediol. So they're regioisomers. They're the exact same chemical formula, but they differ in where they have the hydroxyl group. So 1,6-hexanediol has the two hydroxyl groups on the terminal carbon atoms 1 and 6. So, and by contrast, 2,5-hexanediol, despite having the same exact chemical formula, has the hydroxyl groups as secondary alcohols on the 2nd and 5th carbon atoms. Now, this is very nice because these are useful. These actually melt these structures, okay? And one of them melts and the other doesn't. So let's look at an example of how these chemicals can melt in living cells structures that rely on domains of low sequence complexity. So what we're doing here is we're staining with an antibody to an RNA binding protein called TIA1. So here are these RNA granules in the cytoplasm. And it's really important. These are very important for the control of localized translation. So these are important structures within the cell composed of RNA and many RNA binding proteins. And we know that there are low complexity sequences that are very important for the formation of these structures. So if we add 1,6-hexanediol to the cells, these melt. They're gone. They disappear. By contrast, if we add the regioisomer 2,5-hexanediol, nothing happens. So very, very similar chemicals. One melts these structures, the other doesn't. Let's go now to inside the nucleus. Cahal bodies that are labeled by a protein called coilin. If we expose the cells to 1,6-hexanediol, the coilin or Cahal bodies disappear. 2,5, they don't disappear. So active chemical, inactive. And finally, the splicing machinery as visualized by an antibody to a splicing factor in the nucleus called C35. Those splicing factories are melted by this chemical and not by this. So why am I telling you all this? Well, I'm telling you this because if we go in our test tube now and ask how do these two chemicals work on our gels, the active one, the 1,6-hexanediol, melts them, but the inactive one doesn't. So we have a correlation between the test tube and the cell that causes us to think maybe we're right, maybe we're barking up the right tree. And so now, I don't have time to go into this, but you want to know how does this chemical melt your structures? And we've published this, solved it by solid state, by solution NMR spectroscopy. What happens is this aliphatic alcohol, 1,6-hexanediol, it's two alcohol groups, it's OH groups or hydrogen bond donors that can form hydrogen bonds, consecutive carbonyl oxygens that are hydrogen bond acceptors. So if this binds in this position, which it does, it displaces the Pauling hydrogen bonds that are necessary to hold the two together. Okay? So that's how this chemical works. And the reason that 2,5 doesn't work is because the hydroxyls are not in the right place. These are separated by the appropriate distance between two carbonyl oxygens. In 2,5-hexanediol, the oxygen is here. The hydroxyl group is here and here. Those are too close together to simultaneously bind to two carbonyl oxygen atoms. So we have a very good understanding of how things work. And we were ready to publish this. And I said, okay, we've got to do a control experiment. And the control experiment is if these melt everything in the cell, they're garbage. They're a blunt instrument. And so we put this 1,6-hexanediol on the cells and asked, does it melt actin polymers? Does it melt microtubules? If it melts these things, it's not very useful. So we tried actin, and it didn't melt. We tried tubulin, it didn't melt. But when we tried intermediate filaments, much to my surprise, this chemical melted intermediate filaments. So that's one of the cytoskeletal structures, and I was thinking, oh my God, I don't like this. This is not good. So let's see if I can. Okay, I'm going to show you, if it works, a movie. Okay, so we put it on, and watch what happens to the intermediate filaments. They dissolve right in front of your eyes. And so intermediate filaments are clearly dissolved by this chemical. And let's go over to the control compound, which is 2,5-hexanediol, which we know does not bind to the carbonyl oxygens of the polypeptide backbone. It doesn't touch intermediate filaments. So these follow the rules, but wait a minute, I'm not interested in intermediate filaments. Those are the most boring proteins known to man. I wasn't going to study those. And yet, lo and behold, intermediate filaments use low complexity domains. I didn't know this. But the prototypical, and the human genome, we have 75 different intermediate filaments that we use in our biology. The prototypic intermediate filament has a long alpha helix that allows for the formation of what's called a coil-coil. This is what Francis Crick got his PhD thesis on, studying these in horsehair. And so you can go from this alpha helix to this dimer, and you can even get to tetramer, but you can't build a filament that has 40 subunits without this head domain, the first 100 amino acids of the protein. And that's a low complexity domain. So, we knew for the first time, how are these being melted? They're being melted because the hexanediol interacts with the head domain and disrupts its interaction. So now we have a really good way to work on things because we can make these filaments with recombinant protein in a test tube, so we can study it all now functionally. So here's how we went about things. So I'm asking, how do we know if labile cross-beta structures are of biological significance? So we collected solid-state NMR data on the head domains, these low-complexity domains that form polymers that phase separate, of desmin and neurofilament light. So we can make polymers of this head domain alone, and we can get the spectra of that. Then what we can do is use intine ligation to segmentally label both Desmond and NFL, these intermediate filament proteins, such that the head domain is labeled with isotope, but not the rest of the protein. Now we can assemble these intermediate filaments, and now we can perform solid-state NMR spectroscopy and ask the question, do we see the same spectra? This is our cross-beta spectra. And the question is, when you have the proper assembly, do you see that? And this is a really good experiment, and I'll say this only for the students, because it had 3 outcomes. 2 of them, I'm dead. I kill myself. Okay? The first one, if this spectrum is different from this, I'm dead. Everything's dead. So just close the lab down, Steve. The second one is if there's no spectrum, then I'm dead. The only way I win is if this spectrum is the same as this spectrum. That's the only way I win. So you have to do experiments where you put the gun to your head. That's what you do in science. You put the gun to your head, and 2 chances out of 3, you're dead. Now, luckily, I wouldn't be here if I'd be dead. So the fact of the matter is it worked. And so here's the experiment. So we can make our hydrogels that we could do beautiful solid-state spectroscopy on this head domain alone. It does just like Fuse. It makes cross-beta polymers. And so we can get a solid-state NMR spectrum. We can identify amino acids. Now what we can do is, using intine chemistry, we can label with N15 C13 just the head domain alone, and use the intine chemistry to fuse it to the rest of the Desmond protein. Now we make a real intermediate filament, 40 subunits, everything, gorgeous. Now we take a spectrum of this. This is the real deal. And the question is this spectrum the same as this spectrum? And if you overlay them, they're right on top of each other. So this was the first time I knew, Steve, we're right. This stuff is right. We've gotten it right. And that largely came from pharmacology. So finally, okay, one more point. About a year ago, so we published these data maybe 2 or 3 years ago, but a year ago, a terrific cryo-EM tomographer by the name of, I forget his name. I'll think of it in a second. Working at the University of Zurich, the biochemistry department, they published this paper. I saw this on bioarchives. And for the first time, they got a structure of an intermediate filament protein. I thought these had been solved, but they hadn't been. This is the first ever structure. His name is Ohad Medalia. Anyway, gorgeous structure. 1, 2, 3, 4, 5 sets of alpha helices. Each set of alpha helices has 8. So there's 40 subunits. So here's the alpha helices shown here. And then the head domains protrude into the lumen to hold the filament together, and that's where these cross-beta interactions happen. So when I saw this paper, it was like Christmas. It was just phenomenal. Okay, that's pharmacology. Now, what about genetics? So there's terrific genetics, human genetics on. So what you want to know is, are there any mutations that cause disease in these weird proteins? And the answer is yes. And the mutations are weird. They're unusual. I call them idiosyncratic because they're not mutations that are kind of peppered all over the protein. They're recurrent in the same residues over and over and over. So that's weird. Usually when you think of mutations, you can mutate a protein anywhere. But these mutations are always in the exact same residue. So proline 8 of the neurofilament light head domain is mutated over and over and over, and it's incredibly penetrant. If you have this mutation, you're going to have CMT disease, Charcot-Tooth-Marie disease. And the idiosyncratic thing is not that it's just the same proline, but it can be changed to anything. It can be changed to leucine or glutamine or arginine. It doesn't care what it's mutated to. It mutates to anything and gives the same disease. Same at proline 22 in the NFL head domain. Same with proline 301 in tau to cause Alzheimer's disease. Proline 298 in HNRPA2 causes neurodegenerative disease. proline 183 and Emeron and TFG and MECP2. So there are mutations in these things, but they're weird. They're always proline. Why in the world is it proline? Why, so there's got to be something here. And we looked and said, okay, where are these mutations that the human geneticists had mapped? And this is in the NFL protein. So here's proline 8. It can be changed to any of these. Here's proline 22. So here they are in the head domain, in the low complexity domain. And the geneticists had already done this. They'd already shown that really fouls things up. I mean, so proline 8 changed to glutamine, your intermediate filaments are a disaster. And it's dominant. It's only one of the alleles, and it's a gain of function. So I'll explain that in a bit. So let's look at proline 22 to serine again. So they nailed it. They showed us where they were. Well, where is that? Well, guess what? It's like right on either side of the cross-beta forming region. So here's the cross-beta forming region, and here these bracket it. They're dominant. They're always at one of these two positions, never anywhere else. And mutations in either proline can happen by change to any other amino acid. So why is this? And so here's what came to mind immediately. Proline is the only amino acid that cannot do Pauline hydrogen bonding. It's the only of all other 19s can do it, but proline can't because of the nature of the proline side chain does not have the peptide nitrogen necessary for this hydrogen bonding. So when you have a proline, you can't do this. So the idea was simple, that when you got to the end of this, you insulated it to restrict the cross-beta forming region by providing an insulator to stop it at that point. Now, what would happen if you change to any other amino acid? Well, you change the side chain, of course. This is just an example of changing that proline 8 to leucine. You change the side chain, but at the same time, you reintroduce the ability to form a hydrogen bond. So the zipper should have stopped there, but now it can proceed. And so the idea was maybe the cause of the mutation is allowing that interaction to spread further than it should and create a stronger molecular structure than biology wanted. So now we can do, this is the fun of science. When you kind of put things together, you say, oh, I can do the experiment. So here's the experiment. And this is fun. So here's normal, okay? Normal, it has a proline there, and you stop the zippering of the Pauline hydrogen bonding at this point. Now, let's say you change that to leucine, and this causes disease, and then you look at this, and you say, well, is it this side chain, or is it this hydrogen, okay? And our idea would be it's this hydrogen, because you're allowing it to zip further. The molecular biologist would think, oh, no, it's got to be the leucine side chain. And so any chemist would say, oh, that's a lot bigger change than this, right? Even Pete would probably say that the chemistry there is much different than this change. So, but you can ask. And so how can we ask? So if we've changed proline to glycine, we don't have the leucine side chain. So we lop that off. It's gone. And now what do we have? We have a hydrogen. So we've sustained this. We've gone from the proline that stops it and provided a hydrogen there so the zippering could continue. Or the alternative is let's get rid of that hydrogen by putting an ester. And so you have no hydrogen bond there, but you maintain the leucine side chain. So if this guy is bad, then the culprit is leucine. If this guy's bad, the culprit is not leucine, that leucine side chain. The culprit is having an extra hydrogen bond. So you just do the experiment. And I had hopes that we would know what it would be, but here's how you do the experiment. So you again, use synthetic chemistry, and here's again, I wouldn't have been able to do this experiment had I, as a young scientist, not paid attention to Richard Lerner and Pete Schultz and Ben Cravat wanting to put chemistry into my science. I wanted to do this. So by the time this happened, I'd committed to the same strategy that you had at Scripps, so I could do this. And so here's the experiment. I can do a synthetic peptide, so I can put leuceic acid and have an ester at that bond. So I can do all this, and Now I can ask, does my intermediate filament work or not? And so here's the wild type, makes beautiful intermediate filaments. Here's the human mutation, you changed it to leucine, it's a disaster. Now, here's the variant that has the ester, has the leucine side chain, but does not have the hydrogen. It makes perfectly good intermediate filaments. Whereas if we change to glycine and remove the leucine side chain, it's a disaster. So the problem is you don't want that extra hydrogen bond. You don't want to allow the zip ring to extend. So you want these to be weak. And so this is all over our cells. These things are going on all over the place, in thousands of places. This is going on, and it's balanced just right. And here, human genetics is what taught us that we're on the right track. So let me give my summary take-home points before my acknowledgments. And let me start by saying low complexity domains self-associate via structurally specific cross-beta assemblies poised at the threshold of thermodynamic equilibrium. So this is a really cool discovery. This is true. And we stumbled over it. We found it. And it's happening everywhere in cells, so it's really fun. LCD self-association is widely regulated by post-translational modification, 75 or 80% of all forms of PTM. So this ability to do these things is going to be regulated in very sophisticated and cool ways. Autosomal dominant disease-causing mutations within LCDs manifest by enhancing the avidity of otherwise labeled. So these mutations are a gain of function. They're making it work too good. It doesn't want to work too good. It doesn't want to be too tight. It wants to be just right, like kind of Goldilocks. And the final thing is this weak and transient form of protein self-association facilitates innumerable forms of dynamic cell morphology. So if, we've just scratched the surface. The next 10 or 20 years, people are going to find this going on all over the place. Cells are much more labile and interesting and cool than we ever imagined. But we couldn't touch it. We didn't know it. Now, the next 10 or 20 years, this is going to be all over the place, and it's going to be exciting and fun and offer something wonderful for young students to work on. And I'll close by acknowledging the fact that I couldn't have done this save for the fact that I've had a wonderful group of people working with me for the last 15 years, listed here at UT Southwestern, wonderful collaborators, Betsy Goldsmith, David Eisenberg at UCLA, Rob Tico, terrific SNR, solid-state NMR spectroscopist, and on and on. So let me close by thanking you for listening to me and acknowledging the fact that I'm very honored to have been selected to give this John Martin Memorial Lecture. So thank you. Okay, good.
Thanks, Steve, for a fantastic talk. I should have mentioned that I think I ended up in La Jolla because I grew up on the swampy part of Texas in Houston and came and saw the ocean here. And I should also mention that Tularica is actually one of the places when I was interviewing, leaving academia and looking at industry, super excited about what you had started there. Everyone, I'm Arnab Chatterjee. I'm Vice President of Medicinal Chemistry at the Caliber Skaggs Institute of Innovative Medicines. And it's my honor to be part of the symposium today, welcoming you all, and in a few seconds introduce Gregg Alton to give the next speech. I've had the pleasure of being associated with Gilead now for quite some time. When I was leaving graduate school, I had the pleasure of interviewing at Gilead. I won't give you the dates, but early 2000s, and had the pleasure of meeting several people at Gilead, of which probably the most exciting part to me was John's vision of not only doing great science. I had read his papers of creating entecavir as an HBV drug when he was at BMS and continuing that legacy, but also the amazing teams that he had put together of people of various different disciplines to bring them together to be able to not only do great science, but to impact human health. And Gregg is certainly an integral part of that. I think that inspired me then to come and work with another visionary in chemistry, certainly in Pete Schultz, and certainly what we've been proud of being able to do at Caliber over the last 13 years. So with further ado, I want to introduce Gregg Alton. So Gregg was an undergraduate at Cal Berkeley and did his law degree at Stanford University. Gregg was at the law firm of Cooley for a few years before heading to Gilead. He had a tremendous career of nearly 2 decades plus at Gilead, again, serving as multiple roles within the legal framework, including Chief Compliance Officer, General Counsel, as well as interim CEO when John had left that role. So Gregg has served the institution and the organization in many different capacities. I think he'll talk a little bit about the work today, but again, I've had wonderful interactions with Gregg. We discussed long-acting entecavir prodrugs for treatment of HBV, and I was truly astounded by his ability to not only understand the science of how important entecavir is for HBV therapy, probably not more than TAF, but at least on the list, and certainly the way that it could potentially impact patients in low and middle income countries. So with further ado, I'd like to welcome Gregg to the stage.
Well, good afternoon. This is on, I think. So I'll start up. Yeah, so I was at Gilead for a little over 20 years. Spent a lot of time working with John, so it's really great to be here today. And I actually did not know as well that it was John's birthday, so that's a special honor. So really thanks the Scripps Institute for having me here. I want to give you a little bit of a warning. I've given a presentation to describe Gilead's Access program probably 100, 200 times in my career, but I haven't done it since I left Gilead. I definitely have not done it since John's passing, so it's been a while since I've given this presentation. I've also never given a presentation really focusing on John's role, and I'm going to try to tell a story. So I'm saying this, so I hope you I'm going to apologize in advance if I have a hard time getting through this. But anyway, I'm going to talk about John's role in what we call the Access program. So first I want to talk about John's perspective on medical needs. So he really looked at things in 2 ways. He looked at the scientific contract that he has, or we had as an industry, really driving innovations in therapy and infectious disease. And for John, this was his career. This is what he did. I think there was a lot of talk early about John's early role when he was at BMS, and then he was at Syntax as well, but then what he did at Gilead. And obviously the need for capital. Before the meeting, we were talking about the challenge today in raising capital, and that is really a key critical component of what we do, and the role that intellectual property protection plays there. But mostly what I'm going to talk about today will be John's, what we call the social contract. What we do in life sciences is different than any other industry because we're innovating, it's capital intensive, it requires lots of investment, take lots of risk, but we're doing something that's very, very human. That we're creating medicine, we're creating products that mean life or death for people, or they mean a good life or a challenging life for people. So there's a added responsibility that we have, and John really recognized in the work that he did. And I hope I can really talk about that today. So he really talked about the need for us to not only be innovative in what we do with our science, but also really be innovative in sustainable solutions, because not everybody's going to benefit from that science if we rely on the health systems as they exist. We need to challenge those systems. He recognized that we had lots of scientific progress, and we're really talking antivirals here. However, if you really go back to when I started at Gilead in 1999, there were serious challenges in delivering that treatment in the areas of HIV and hepatitis, but other areas in the developing world. And so John really took it as we need to have that scientific contract, but likewise, we need to have the social contract in terms of what we do, because if we're going to be innovating science to benefit humans, we need to make sure that humans get that science. So this will be my only scientific slide or semi-scientific slide that I'll have. So when you talk about the scientific contract, I'm going to be very simple here. John had a real vision of delivering simple, tolerable, and effective therapies. First off in the area of HIV, but also in hepatitis B and further in hepatitis C, and beyond that. So if you look at the, I think there's a pointer here, but if you look at the hand there, the handful of pills, this was in 1996 after the protease inhibitors came along. And with the protease inhibitors, you could have heart therapy, and so you could actually do a pretty good job of suppressing the virus in an HIV-infected individual. But there were two problems in 1996, and John was already working in HIV at this time, obviously. Number one was resistance, and I remember early on in my career at Gilead, everyone was talking about resistance, the different type of resistant mutations that you'd get on this drug or this drug, and I think the physicians, the big challenge for the medical profession was, which drug to switch to next, because you would put someone on a drug, you would monitor the resistance, you'd develop a particular mutation, then you'd have to take a different drug. The other thing that was going on in 1996 is the complexity of the regimen. Patients would have to take multiple pills multiple times a day, some with food, some without food. So for a patient who's really, really, really diligent, can really, really follow that, that's just a huge impediment in your life. For many patients, they just could not do it. Many patients could not follow that. The other thing that in 1996, in addition to the resistance and in addition to the challenging regimen, was the toxicities of the regimens at the time. So you could be virally suppressed, and if you're really good at structuring your life around your medication, you could keep up with the complexity of the regimen, but you didn't feel good in many ways. And all the different drugs had a different side effect. There were CNS toxicities, there were gastrointestinal toxicities, there was neuropathy, all kinds of different challenges that patients face. So in 1996, you could virally suppress the drug, the virus in a patient. A patient could actually look at living a relatively long life, but it was not the life that they wanted to live. So in addition to the medical challenges. So John's vision, and I am absolutely 100% sure, this is a vision John had very, very early on in the HIV epidemic, was coming up with a single tablet regimen that was safe, tolerable, and effective. And so you look at the advancement across time. That was John's scientific contract. And the same thing he did in hepatitis C. I think John, we were working on hepatitis C very, very early on. There were some intellectual property challenges early on that many people who have been in the industry for some time would recognize. But we were able to get back into hepatitis C later. John was also very big on not just building at Gilead. We talked. Thomas talked about the collaboration with IOCB Riga. With hepatitis C, was really looking at Pharmasset and the work that was done there, and bringing the molecule in, and then building around that to get the regimen. So we got to the therapies we have today. So I want to turn to social contracts. So I mentioned, John had this scientific. We developed this single tablet regimen. We had durable product that had very low levels of resistance, very tolerable. But at that time, so if you go back to 2001, there were a lot of challenges to access to medicine. And I remember, I'll try not to tell a lot of stories, but this one's really, really vivid for me. In 2001, right after VIRID was approved, I want to say it was like a week after at most, I'm sitting in my office, and I think it was on a Friday afternoon, and John comes in my office and he says, Greg, we have this product, VIRID now, of course, because that was the big excitement that we finally really hit one. But he's like, with VIRID, it's very tolerable, it's very safe, has a high barrier to resistance, very simple for patients. We need to find a way to get this into sub-Saharan Africa and other places around the world that are hit so hard by HIV. And he said, I want you to, and I was general counsel at the time, but there was really no one else that was sort of in this role. He said, I want you to find out what are the other companies doing, what's working out there. And so over the weekend, and for the next few days, I did as much research. This was really before we had high-powered Google and stuff like that, but I was able to look at what some of the other companies were doing, and he came back to my office probably Monday or Tuesday of the next week and said, so what'd you find out? And I said, John, I don't think really anything's working right now. And I said, as far as I can tell, all that's out there right now are a lot of PR statements about commitments to doing things, about companies that are there to fight HIV. But I said, John, I can't see any evidence that there's any concrete programs out there. I can't see any evidence that actually large quantities of medicine are actually getting to patients in sub-Saharan Africa. And what we saw at the time in terms of what the challenges were, so this is no criticism of the industry. The industry was really just waking up to this challenge. There were numerous challenges, delays in regulatory approval. So you were dealing with countries that just did not have robust regulatory agencies. We dealt with taxes and duties in countries. Just if you were trying to donate drugs, it was a challenge at the time. Forecasting, so what is the demand? How much drugs should you make? And I mean, Tyann, all my colleagues from Gilead are here, or my close colleagues are here. Remember, trying to figure out how much drug do we want to make? Because you don't want to make more than the demand's there, because that's a burden on the company. Healthcare workers, shortage of trained healthcare workers. When you get into the rural communities, in particular, how do you have a physician who understands how to use the products? Political commitment, I'll come back to political commitment later, but this is a big challenge in terms of going into countries and asking, we have these products, can we work with you to get these available to your patients? And there just wasn't, in many countries, a huge drive at the highest levels to do that. Treatment-related cost to patients getting to healthcare facilities, the pricing of ARVs, the cost to healthcare systems, the cost to patients is out of pocket, limited distribution networks, and then just limited healthcare infrastructure. So these are all challenges that we were facing at the time. But in John's world, you just take on these challenges. And I think for John it was, we're not gonna back away. We need to figure out how to do this, and we're going to come up with our best ideas, our best hypothesis. We'll spend some time, and all my friends here in the room, we worked on this, and we'll come up with our best approach possible, and we're gonna try. And I always think of this as sort of a science. You come up with your hypothesis. Well, you have a problem. You come up with your hypothesis. You come up with your best idea how to solve it, and then you go forward and you see what happens. That's the experiment. And John always actually talked about this as the great experience. So on April 4th, 2003, Gilead announced its first access program, or it's an access program. And I want to just point out a couple things on this that I'll touch on. So it covered about 15 or so countries, no profit. So we basically would make our product available at our cost of manufacturing. And the idea was to be sustainable. So we didn't want to make money, but we didn't want to lose money. We just would have this as a program that would be sort of neutral to us on our revenues. The program would receive requests from the drugs in 68 countries, review requests, and ship the drug directly to the countries. And our thought was, there's such a need here. There's this big emergency. Let's just send it, get it to people who want it when they need it. So that was the thought. We also partnered to look at a demonstration project in Uganda where we underwrote clinic activities. This is dealing with some of the healthcare worker shortages or needs. We donated the drug. We actually did have to deal with taxes and duties to get it in, but this was our first experience there. Really to show that you can use HART in resource-limited settings, particularly when you have a product like Gilead that addresses a lot of the challenges that were there. We demonstrated rapid scale from 100 to 1,000 patients, and we were really looking for this to validate and support additional funding from the Global Fund for this type of work. I would say the first attempt, our first experiment failed. So again, but there's a couple reasons for this. First of all, the direct import. Directly importing the drugs was very, very cumbersome. It was very difficult, particularly when you're dealing with chronic therapy for patients. And we heard this loud and clear from organizations like MSF and others who said, this is just really hard for us to administer this program. We did not think it would be necessary to get in the WHO essential medicines list because we thought we're dealing with HIV here. We're dealing with a drug that is, by scientific standards, clearly in great need in these countries. So we thought that would be a process that would just be too much time. That was also a mistake because we learned later how important it is for countries to have that WHO stamp of approval. Lack of medical education. We also did not, we underestimated the need for different countries to understand what Gilead would do. I think that we In the United States and in Europe, I think the information, the science was quickly absorbed within the medical community. I think, Clifford, I think we can remember, we had an example in I think it was Jamaica, where we shipped, I want to say like 30,000 bottles to Jamaica, and then nothing. And there was no further orders. And so we looked into it, we said, what's going on here? And they said, we don't know what to do with this. We don't know how to use this product. And we're like, oh, so that's the sort of light went off there. Forecasting, I mentioned forecasting as a challenge. We just weren't doing a very good job forecasting at the time. And we actually found ourselves writing off product that would expire because we just didn't have the patience for it. So we weren't sophisticated there. And finally, price. So at the time we launched Gilead, at cost, I think it was roughly $17 a month for Gilead. But what was being used in the developing world were some of these older regimens using D4T, AZT, and lamivudine that was very, very cheap. The drugs didn't work that well. The combinations weren't very good, and they would obviously use a protease inhibitor or a non-nuke as well on top of that. But because Gilead was at a higher price, governments just weren't willing to use it. So based on this, John really took all this information together. He looked at what we're doing, and again, I talk about this sort of being the experiment. Well, the first experiment failed. That doesn't mean we give up. We learn what we learned from that first experiment, and we try again, and we keep trying. So you see in 2003, we did this with no profit pricing, direct shipments. We got up to about 30,000 patients out of roughly 40 million who needed the product at the time. So that's why I say you can call it a failure. It didn't work. We didn't meet our expectations. So in 2006, John really insisted that we change how we're doing this, and we did a couple key things. So we established our international access operations, and this is when we brought Clifford to lead that. And part of this was also at the time before 2006, our access program was running within our commercial group, and the head of the commercial group at the time was a guy named Kevin Young. And I remember sitting with Kevin, he's like, Greg, this is really, really complicated. He said, this is really not what we do in commercial. And so he actually said, I think we need to separate this out. And he actually went to John, and John absolutely agreed with that. So we brought Clifford over, we separated it out from a commercial, we gave it its own sort of direct influence. We established a broad distributor network. So we worked with companies in all the regions throughout the world. So these are local companies in Africa, probably about 5 or 6 in Africa, in Latin America, and Southeast Asia. We provided them with a lot of regulatory training, a lot of safety training, so they could actually do all the work that we needed them to do. Because we recognize we need somebody on the ground in these countries, and we can't really build our own infrastructure and keep our costs down the way we needed to. We had broad registration, and we actually not only committed to registering in all the countries, we actually put on our website a list of all the countries and what the status of the registrations were, so people would see that we were working on it. We had a dedicated medical affairs group within the access operation, so these were physicians that we would actually send around the world to conduct medical education training in all the markets, so that the clinics that were needing the drug would also know how to use the drug and the value of the drugs. We developed supply chain expertise, and I'm going to the essential medicines list. The other one we did is is generic licensing. So in 2006. I mentioned before, John said, what are the other companies doing? We also began to recognize the only companies that were really doing a good job in places like sub-Saharan Africa were the Indian generic companies. They were there, and they were delivering large amounts of what we considered the inferior products, but they were delivering the products. And so, John said, why don't we work with them? Like, why, if they're able to do it for D4T and for lamivudine, why can't they do it for Gilead and eventually Truvada and the other drugs that we would produce? And so I said, okay. And then in typical John fashion, we flew to India, and some of my colleagues here were with us. And I remember really, again, sorry, another story, just this is one of the vivid ones. John was also very hands-on. He was not someone who just sat in his ivory tower and talked about things. He wanted to be there. He wanted to see things. We flew to India. John was with us, and I think we were in Hyderabad at the time. And I remember being in a little cafe on the side with one of the Indian companies and the senior team from the company. And it became very clear right away why this was going to work. Because the senior team at this is Mylan, I believe, at Mylan, They were all medicinal chemists. And so seeing them with John talking was actually really, really amazing, because they weren't talking about, how we're going to do a deal or stuff. They were talking science. They were talking about how they were going to work to get the cost of Gilead down, and what kinds of things they could do with the process chemistry, with the other steps to bring that down. So we launched that in 2006. And the idea was also, we wanted to do this in a way that would take advantage of what's already working. And I think my next slide gets into this. Actually, not quite yet. So we're going to move ahead. So we wanted to get into to what was already working with the generic licensing. So if you look at we wanted to replicate a generic model. So we wanted to have multiple companies competing. So we believe that the competition was going to be critical to making this work. That was one of the key aspects of it. We wanted to have no price controls on our partners, so that they would be freely competing for the marketplace. We wanted to be able to sell API to each other. We also recognized that ultimately there'd be major producers of API, other companies that would be just making the tableting, but we wanted to have that scale of the API. We wanted to, we gave them a technology transfer of how we were manufacturing it. I know Tyann and her team spent a lot of time doing technology transfers for us. But ultimately, we wanted them to actually develop their own processes, because we really felt that that was their expertise, was in how to drive down cost of manufacturing. And with that, we also wanted to be fairly hands-off in what they do. So we felt that if we were going to be putting a lot of control over their manufacturing, whether it be on their quality or other things, that would disrupt what they are doing. So we really wanted that. We said, really what you need to do is have quality standards of WHO pre-qualification, EMEA, and then we also had tentative FDA approval under the PEPFAR program at the time. So that's what we required, but we were not sending our people in to monitor them. So that was the generic model. I want to back up now. The other thing we did in terms of our tiered pricing, and I think this is important, is we didn't just look at G&I per capita. So we did a tiered pricing model where for the lowest income countries, it was at no profit. For middle income countries, or those that had a moderate level of G&I per capita, we would price with a slight profit, not a huge profit. And then for upper middle income countries, we would negotiate prices. So for example, Brazil or China or other markets, we would negotiate prices that would be significant discounts to the US, but reasonable for their access. But for John also, he was also very clear that we can't just look at G&I per capita, because you need to look at almost like G&I like per patient or by the need. So we also looked at disease burden. So, and I'll give you another example of a country, for example, South Africa. South Africa is an upper middle income country, but the disease burden in South Africa was so great at the time, we really recognized we need to do something beyond just evaluating them at G&I per capita, because of the burden that HIV was having on their healthcare system. So we took that all into account in how we tiered the countries. So South Africa went into a low-income market, as did other similar countries. So that was part of the model as well. And if you look at the pricing, this was $17 a month in low income, $30 per month in lower middle income countries, and then as I mentioned, we would negotiate for upper middle income countries. These are all incredibly big discounts to our commercial markets. And I will just now to touch on a little bit also, one of the values of breaking our access operations away from commercial is a lot of the commercial, and I see you here, Melissa, and a lot of countries have the tendency to want to reference these prices. So we had many times where, for example, in the US, you'd have like a health system in the US say, well, why are you making it so cheap in South Africa? Why is it so expensive for us? And John's like, we'll tell them. We're going to flat out tell them, because South Africa is different, because they have a disease burden unlike yours, because they are a much poorer country than yours, and we just have to be willing to have that conversation. But there was always a kind of a strain between our commercial group and our access operations, and John was strong enough to say, we're going to allow that strain to occur, and we're going to make sure our commitment is always to the patients, and we're going to continue doing what we need to do to make sure that product's available to these patients. So going back, I'll pass over this. We're also the first, John led us to be the first company to join the Medicines Patent Pool. So this is a an organization out of Geneva. And I'd actually say that we actually helped form the medicines patent pool because it's a group out of Geneva that really was advocating for licensing. They would actually take a license from us, and then they would out-license to companies. It still exists today, and a guy named Charles Gore runs it, so people who are involved in hepatitis know Charles quite well. But we were the first company to join that pool, and you can see that's Ellen Tahone there. I think that's Philippe. Anyway, but that was part of John's vision to do that. When I say that we basically formed it, they basically took our form of contract that we did with our original licenses, used that as the basis for the first one. Now, it's evolved a lot since then, but that was part of it. So over time, our access program evolved. The generic licensing expanded to a number of other countries. So now, this was probably as of 2016, include all of Africa, but you can see it includes parts of Central Asia, parts of Eastern Europe, and Central and South America as well. In terms of what our generic licensing was able to do, you can see that in 2006, the cost of the products were, so that's Truvada and Gilead, $26 and $17. Brought it down to $1.80 and $3.70 in 2018. I think it's actually even come down from that. But if you think about, and I'll show you some slides later on this, the benefit that this has for organizations like the Global Fund or PEPFAR and be able to scale up antiretroviral treatment, I mean, just means you can treat a lot more patients. Also, it just takes away the temptation of organizations to use an inferior drug that's cheaper with the idea that they don't want to deny anybody access to medicine. So we were really working hard to get these prices down. So this is another slide that I know it's a little bit busy, but really shows some of the factors that went into it, because it wasn't just the generic licensing, but it was also the global fund. So if you see, you can see the price of the drugs going down, but you can also see the funding sources going up. So the global HIV funding going up. And this all was happening at the same time. So you had funding coming in, you had prices coming down, you had volumes of patients going up at the same time. And I think those were all related. So this is my way of saying it's not just Gilead doing this. This was the investment of the global community, also lots of NGOs, lots of organizations around the world participating in this activity. But I will touch on this slide a little bit later, including global reach. So this is what we were able to do. I mentioned we were very not satisfied with our program in 2006. It was about 30,000. By 2018, we're reaching about 12.5 million. I think Tomas's slide had us at 16 million, I think, as of today. So it continues to grow. But this was very, very successful, what John was able to do here. But I would say that and this is as of 2016, we also recognized in 2016 tremendous unmet need exists. So I pulled the slide from UNAIDS from 2016. So in 2016, you were still getting about 5,000 new HIV infections a day. That's a startling number. 64% in sub-Saharan Africa, about 400 among children, high rates among women and young people as well. So in 2016, we were actually pretty pleased what we were doing in the treatment expansion, but obviously we weren't, completely happy with the way that HIV was going. If you look more about what's going on in terms of need, you had 36 million people living with HIV. You had, look at the new infections, 1.8 million per year, and AIDS-related deaths, in 2016, about 1 million. So it's sort of a glass half full, half empty. And I think for John, it just meant there's a lot more work that we needed to do at the time. So a couple things that we launched around this time. I think Tomas mentioned lenacapavir, but PrEP was coming along around this time. So PrEP is using antiretroviral therapy as a pre-exposure prophylaxis. So we really saw that as something that could really address what we're seeing, which was we're getting treatment out there, but there still are far too many new infections. So we looked at different programs. We had a program, for example, in Brazil, making PrEP available in referral centers. And I think now Brazil has wide access to PrEP. But it was programs like this that really got it going. I think John recognized that we need to get people the experience with PrEP, because at first, PrEP was actually quite controversial. People thought there'd be disinhibition or that it would be too expensive or other things. I think people, once they started using it, got very comfortable with the concept of PrEP. The DREAMS project, this is something actually Clifford really championed in his role. And I'll show you some more data on this, but one of the drivers of HIV in Africa is young girls and older men, and the passing of the virus in that context. And so the DREAMS project, and Debbie Birx was actually one of the big champions of this when she was at PEPFAR, was really targeting young girls and young women for PrEP use and see if we can curb that epidemic. So that was very successful. And then getting PrEP around globally, just filing for approval, and then increase the medical education around PrEP. And I think so right now, you can pretty much eliminate the risk of transmission or exposure to HIV by taking 1 pill a day. In about, a month or so, lenacapavir is going to come to market, and then with 1 shot every 6 months. So it's an injection every 6 months, pretty much 100% effective in preventing a person from becoming infected with HIV. So this is a huge opportunity. Obviously, with the 1 pill a day, we saw it then, and now the science has continued to evolve, and so now we're going to have a PrEP that's 1 shot every 6 months. The other thing that we were working on is rural communities, and also how to reach men as well. So here's a project that we did in Tanzania, and I don't know if people here who know Yup Longa, a very good friend of John's. They were very close together. He was actually the one who set this up, and he set this up in a model where you go into a rural community in Tanzania. I've actually been to this clinic recently, and they're doing really, really well right now. And we built up their capacity of having a central laboratory in this very rural community. We funded some lab technicians, and then it had a hub and spoke model. We have clubs out throughout the community that were support clubs, essentially for people to be adherent to HIV therapy, to get everybody in the community tested for HIV, and to engage in PrEP. So PrEP is happening there. So that was a part of that commitment to help, again, continue to curb what we saw as a lot of new infections. And the idea around the Tanzania project was test and treat, with the idea that if you could get a significant portion of a population tested, screened, and onto antiretroviral therapy, you'll suppress the virus and they become untransmissible, so that you can prevent infections by treating the infected. So here's a slide, and I have to say John would be both proud and concerned of this slide. So this is showing what the impact has had in terms of averting AIDS deaths by the introduction of antiretrovirals. And if you look at remember the slide that Tomas had, you can see that period from like 2008 to 2016. Those are the ones that we're talking about, the ones that Gilead developed, most of them with some sort of tenofovir backbone in them in various forms. You can see about 21 million AIDS-related deaths averted through the use of antiretrovirals. And this is because of the science of the companies like Gilead. It's because of the commitment of people like John to make sure that those products can be available to people where they're needed. And it's also a testament to Global Fund, PEPFAR, and the work that the NGOs are doing around the world. John would be concerned, and this is where I will try not to get too political, but if we back off from the funding in the area of HIV globally, the concern that a lot of people has is that that blue line is going to start coming back up again. And because I mentioned earlier, this is I'll come back to a prior slide, lack of political commitment. It might be easy to say, well, the countries need to take over these programs. The countries need to take over the providing of antiretroviral therapy. They need to take over the medical education programs. They need to take over the PrEP programs. The reality is, and I've spent a lot of time on this, and I'll talk about hepatitis C in a second here, they won't. A lot of these countries won't do this. The political will just is not there. And so I think it's a situation, I think John recognized that we need to really stop being, us, them, us, our country, their country, and just be human and be there to address the problems for people, as best we can as a company and as best we can as a country. John actually was on the President's Advisory Council on HIV and was very, very vocal about the need for the U.S. to continue to fund these programs. And I think Pete said if John was here today and around, he'd be probably pretty adamant and pretty vocal on the need to continue programs like USAID, like PEPFAR, and other programs that mean so much to these countries and have been so successful. And there's such a risk that this could turn. So I want to talk a little bit about John's impact today. This is a very busy slide. I won't read all of this, but I think it's really promising to see that in 2023, 1.3 million people were newly infected with HIV compared to 3.3 million in 95. Again, this is sort of a glass half full, half empty. I think that's a great progress, but we obviously want to do better. One of the stats that really kind of struck me was in 2023, someone dies of HIV every minute. To me, that's startling. That's a startling statistic, but it's clearly a lot of progress, and I think it's the combination of that scientific impact contract and that social contract that's allowed us to get here to work through this. So now I'm going to change gears a little bit. I think that I've talked a lot about what we've done globally in HIV. In 2013, Gilead got involved in hepatitis C when we acquired Pharmaset and then developed our direct-acting antivirals for hepatitis C. Obviously, we really wanted, there's a huge global burden for hepatitis C, but there's one key difference for hepatitis C versus HIV in terms of the health systems, which is there's no global fund. There's no PEPFAR. So all the hepatitis C therapy is either going to be from a local government or it's going to be out of pocket from patients. And so that created a unique challenge for hepatitis C. But the burden was there, and John was absolutely committed to doing what we needed to do. We already had our access program set up, and for better or for worse, many of the countries that are impacted by hepatitis C are the same countries that we're working with in HIV. So we already had a lot of the ground infrastructure there, so we didn't need to recreate a lot. And we also had the very good experience in generic licensing. So we actually announced our generic licensing program for hepatitis C well in advance of the product being licensed in the US. We really want to get ahead of that, let people know this is something that we'll talk about, but we're actually going to do. So those licenses were in place. So we put those in place, but there were a couple differences. And one key difference with hepatitis C, we recognized that there was no global fund and no PEPFAR. And so the concern then was that the Indian companies would not be incentivized to invest heavily into hepatitis C because there was no guaranteed buyer out there for the product. So we wanted to have fewer companies involved, so we actually limited the licenses. We probably ended up having more licensees than we actually wanted to, but so it was difficult to turn many away. And the other thing we did is we raised the royalty rate from 5% to 7%. And the reason to do that is we recognized that we would actually have to invest more into these markets. And we had this conversation with the partners about this. But in John's mind, that was going to be really critical to ensuring that particularly in the area of hepatitis C, where we really need to get people away from monitored therapy. We need to get people to understand that we can really do away with a lot of the diagnostics because their experience was with interferon-based treatments, and as we moved away from that, we could actually reduce a lot of the costs. So we wanted to be able to invest in all those things. So we launched that, and then not dissimilar to what happened with HIV, we saw the prices come down. And again, this is because the Indian companies, what are they really good at? They're really good at medicinal chemistry. They're really good at figuring out how to get the cost down. So we saw the cost going from about 400, 300, and 400. That's 400 for the combination soft lodipisphere, and then the Sobaldi going from 300, then 400, 300 to 300 and 250, and then ultimately dropping down to 19 and 10. And this is because of the competition. I just want to touch on a couple things. I don't have slides for this, but a couple of things that we had ongoing at this time is we engaged in, this was actually something John initiated, 2 big programs that we had for a proof of concept around hepatitis C. And one was with the country of Egypt, where John had met with the Liver Council at an EASL conference. They recognized what was coming in terms of the direct-acting antivirals. Egypt had did have the highest burden of hepatitis C in the world. And we're able to work with them and come up with a program where we could provide them with an extremely discounted product and support for treatment of their entire population. So that's something. They were also able to get some World Bank money, I believe, as well. The other program was also evolved out of Egypt. We worked with the country of Georgia, which also had a very, very high rate of hepatitis C. And the CDC was also involved in that. And we were able to actually provide them with free drug, but they actually invested in education, in stigma reduction, in awareness, disease awareness, screening programs. And these are both very, very, very successful programs. And I bring this up here because with those programs, I think the rest of the world started to see that in these resource-limited environments, you actually can do this, and you can scale this up. And that's what led to the volumes, that led to the cost reductions to where we are today. So by adapting the model for hepatitis C, this is a dated slide, so it's probably 2016, 2018. About 1.7 million were treated with Gilead medicines in low and middle income countries, 1.7 million. About 68% of those were from our licensed generics. And then we had 14 voluntary generic licenses at the time. So again, John's vision, John's, as he calls it, the great experiment playing out again for hepatitis C. We've done similar programs in hepatitis B with, it's been a little more of a challenge in hepatitis B because of the lack of funding and the need for long-term therapy. But this was a model that really worked well for us. So looking at the legacy of increasing access, you can just see, Tomas has a slide with the drugs coming across. This is the slide that we would have showing the timeline of our access program launched in 2006, evolved in 2003, but then really our access operations model coming in 2006, and then moving towards medicines patent pool. I didn't mention Amazon. I'm not going to talk about that, but this is an antifungal product that we actually did a lot of work with. I mentioned the Egypt program, so we became available in there, and we did the licensing in 2014. 2016, we had 10 million on treatment, so that's quite an advance from the 30,000, and then 12.5 million on HIV therapy in 2018. So just looking at John's global impact, I think it's just outstanding. And again, this is a dated slide because this is the data that I have, but about 13 million on people treated on HIV therapy, hepatitis C, 3.5 million treated on soft-based regimens, and in hepatitis B, reaching about 2 million patients, which is too low, but still amazing impact that John was able to have through the programs that he put in place. So I've talked a lot about the developing world, but I also just want to make sure that, John would want me to mention that he was very committed to the challenges in the US, the domestic crisis of HIV. So this is a program, this slide shows graphically where HIV exists mostly in the US. You can see it's largely in the South, and I'll get into some more of the data on this. It's also predominantly in the Black population in the US, and it also has a disproportionate impact on Black women. But the real reason I want this slide is that John, we always have this what we call the waterfall slide at Gilead, and this showed what we estimated, the number of patients infected, number of patients who have been screened for HIV, number on treatment, number of virally suppressed, and you see it go down like this. And ideally, you want it to go straight across. You want everybody who is infected to be screened, treated, and suppressed. But John was frustrated that we weren't able to reach all the populations. And one thing that we were, with John's direction, we're able to do is put together a program for really understanding the epidemiology of HIV. And so this is the AIDSview project. I'll give a flag for that. I'm still an advisor to AIDSview. This is a partnership that we established with Emory University, Jim Curran and Patrick Sullivan out of Emory University. The CDC is very involved in this as well. And this, anyone who's really interested, go onto their website because you can really get a lot of different cuts of the epidemic of HIV in the US in terms of which populations, which geographies are most impacted. So John was, wanted us to do that because what John wanted to do is understand where are we having these challenges? Where's this gap coming from? And so by working with Emory, we're able to really dig into that. And I mentioned Black, so here's one of the disparities that really came out. So if you look at the Black population, 30% of the US population Black, but Black people make up 40% of the HIV epidemic. So that was really startling data that came out. And then if you look at Black women, it's even more startling where the disparity of HIV in Black women. So that was something that was a big wake-up call for us. Another population that really came out is the transgender population. Currently, 4 in 10 transgender women live with HIV. In some populations, in metropolitan areas, you'll see it up in the 60% range of people living with HIV. So, this was really, really critical data for John to say, okay, now we can really focus our efforts. So he was able to really then communicate with our medical teams and our commercial teams to say, okay, how do we get these populations? And so, a lot of things, I don't have slides on all this, but we can change some of our commercial tactics to address these populations. So people who actually watch a lot of TV may see some Gilead advertising that actually targets these populations. That's been very, very welcome by these communities. I can tell you right now, this was something they really wanted us to do, to help out. And then we were able to do a number of other programs as well by having a really patient program. I noticed that trans community comes up twice on this, and it's probably just a typo. But one thing that John also wanted to do once we actually had these data is really get to know these communities. So we spent a lot of time with John himself and the senior teams really getting out to meet with the leaders in the Black community, the Black HIV community. There is a strong presence there, and in the trans community, and really understand what can we do, how can we help you to get more people screened, get more people on PrEP, get more people on treatment for HIV to really curb the epidemic. So one of the things that came out of this is a program that we call the Focus Program. It's on the front lines of the communities in the U.S. And this is really focusing on getting into some of the federally qualified health centers around the U.S. and in other low-income areas in particular that have high rates of HIV, high rates of prevalence of HIV, and high rates of incidence of HIV, and really teach these centers in these communities how to do HIV screening, because a lot of these centers were very, very uncomfortable with it. And we're able to really put metrics around that through this program. It was a heavy investment that John was willing to put forward into these communities to increase the number of people who are able to become aware of both the risk of HIV, but also the availability of PrEP, or if they are HIV infected, that you can get onto treatment and you can live a long, normal life and you can become non-transmissible for HIV. So that was one of John's commitments. And we did something, we also recognized when we launched into hepatitis that we could do something similar. So we also launched a program called Hep Connect, and this was largely focused on the opioid epidemic in the US. So when we launched our hepatitis C products, I mean, I remember there was a huge number of baby boomers who very quickly were able to come through the system and get treated for hepatitis C. And that was phenomenal, that was very, very successful. But even today, there remains a significant population in the US, particularly among those who are hit by the opioid epidemic, that need to be reached. So we launched the Hep Connect program, and this is largely in parts of the country that have disproportionate burdens of use of injection drug use for hepatitis C. We do, again, treatment. We fund screening, awareness programs, and this is in places like Appalachia and rural parts of the country. So again, part of John's commitment. And this was a very, very large financial commitment John put forward. So I'm gonna end by just going back to what we talked about in terms of John's perspective on medical need. And I say this to all of you in the room that might become CEOs somewhere, or that you may run a lab and you're going to out-license product to companies. Try to keep this in mind when you think about what you're doing. You have your scientific contract, absolutely, and you need to innovate. You need to create the products that people need. But recognize that the products don't always get to the people who need them. And there are things that you can do to make a difference. And I think John is a fantastic example. I would also say that what Gilead did here is not something that will work for every disease area and every product. You can't generic license everything, for example. You don't always have a global fund. The challenges are going to be different, but it's your job to really think about how you do that, break it down into its parts, and you got to try. So what would John say? So I think it was it Peter or Tomas who said, what would John say? We say it a lot at Gilead. He's like the North Star still today. It's like, so what would John say if he were here today? Well, the first thing to say is that True scientific innovation in our industry develops drugs that address true and medical needs of patients. And this is I had another John saying, but he got really frustrated when he see Me Too drugs come out. He's like, ugh, another blue drug, red drug. You know, it's like this is no different. It's just like another version of something that's already out there. I understand that that's going to happen, and people, you know, you're not going to toss out research that develops something, and there's a value in having competition, but you know, John was really focused on doing something different. So in an HIV, he didn't want to develop just another, you know, AZT or D4T. He wanted something that would be different, and he took the risk on tenofovir that no one else was going to take to do that. John would also say, just, you know, despite, you know, all the talk about the generic licensing, I think that John was always very clear. Yes, we do generic licensing. It doesn't mean that we're not strong advocates and supporters of a robust intellectual property protection. We need intellectual property to fuel the innovation that's out there. It's how you use that intellectual property, I think, that John would say is important, because, again, coming back to what I said earlier, we're not making nice-to-have products that, you know, you can either have or not have, or, you know, but these are products that are needed by people to live, and you have to have a different sort of business mentality about that. That's something John would say. I think John would say, take time to know and understand the true patient population that your product's going to address. I know that those of you who are, and I'm on boards right now, and I remember Gilead, we look at, you know, what the potential market is for a product. That's great, and I think we should do that. We have to do that to encourage investment. We have to do that to have a sustainable business. But look beyond that. Look at the patients that don't normally fall into that category. Look to the, as I mentioned, like in HIV, the black women or the trans population that may not be the big population, but these are the populations that really need it, in addition to your potential market. Be innovative in addressing the challenges that face patients in accessing medicine. The health systems often fail to reach certain populations, and we can play a role in that as an industry. And I think John would also say, take risk. Don't say, I can't do that because it's risky. I think John would say, we take risk in our scientific innovation all the time. We have to take risk in what we're doing to see that patients get the medicines, even when it may not be the easiest thing to do. So take risk in developing those solutions. And finally, measure your success in broad patient access with a sustainable for-profit business model. I think John always really wanted to talk about how many patients are we reaching. You know, when he went on his earnings calls and things like that, they would talk about the revenues and how much money we earned in the quarter and what our profits were. But what really drove John was knowing that, you know, how many patients are now being treated by Gilead or by Truvada or by Atripla or by Biktarvy or whatever the product was on the market at that time. And then he would go to, but how many need it? And how are we going to get to those patients? So that was really what drove John. And I just would leave it to all of you to try to have that same mentality, whether it's you're starting your own company or you're involved in a company or you're out licensing something, you can insist upon some form of commitment to doing these things, because that is I think what we should be doing as an industry. So that is it. I think I'm right about on time.
Okay, well, I'd like to just finish up and thank Steve and Gregg. I think their talks kind of underscore why I think John is such a terrific role model for people in science and people starting their careers in science, because he really valued, you know, innovative high-risk research, and I think Steve really underscored that. And he also really valued translating the impact of basic science on global public health. And I think Gregg really did a terrific job describing that aspect of John's career. And finally, a little story. I don't think the Gilead people who are here realized I played a major role in the early trajectory of Gilead Sciences, okay? Because early on in my career at Berkeley, Mike Reardon came to me with Peter Durbin. and said, we have this great idea, Pete. We're going to start this company called Gilead, and we're going to make dystomycin analogs that bind to DNA site-specifically and turn on and off genes. And because I was kind of the person who invented all of that in Peter's lab, they came to me and said, we want you to be a founding scientific advisor. And I looked at them, and they gave me a scientific advisory contract. And I looked at it, and then I went back to them, and I said, never in 100 years are you going to be able to turn on and off genes selectively with dystomycin. I know because I made it. It's not going to work. Why don't you go out and find somebody who can make a real drug, okay? And then they went out and they found John, okay? It's a true story. And at the time I was, and still am, highly principled. So I said, I really can't be a scientific advisor and take all the shares you want to give me. And at the time, so I ripped up the agreement and gave it back to him and said, I can't do it. It was 100,000 shares of Gilead stock. And what you don't want to do today is tell me how much that's worth, okay? So that was my first financial decision as an early young assistant professor. And I'll tell you why they don't let me invest the Scripps endowment, okay? Because shortly thereafter, I got a call from my old college roommate at Caltech, who was a physics major at Caltech and failed out of physics. And back in those days, if you failed out of Caltech, you could transfer to Berkeley. So he became a communications major, and he was a terrific person. And shortly after I ripped up that contract, and Joe called me, Joe Lettieri, and he said, Pete, we're raising money, we're looking for investors, I'm into special effects. And would you like to invest in a trilogy we're making, and we're looking for investors because we don't have the money. And we're going to make it as a trilogy to save cost. And I said, Joe, I make $32,000 a year and I can't afford a house, okay? There's no way I'm going to do this, okay? So about 6 years later, I'm in the movie theater and the movie screen's flashing, who did this, who won that? And up comes Joe's picture. And it was who won the Academy Award for special effects in Lord of the Rings, okay? So that was the second thing I didn't do, okay? Anyway, on that note, we'll finish up today. And again, we all remember John, and we, like Gregg said, we all wonder, if John were here, what would he say and how would he help us? So we miss him terribly. And finally, I'd like to have you all join us in a reception outside. Thanks for coming.